Alright guys, here we are going to talk about cancer, what the major causes of cancer, and what happens on the genetic level in order to trigger the development of cancer. So up until this point we've been talking a lot about the cell cycle and how the cell cycle is controlled. We've talked about mitosis, and we can't leave that big elephant in the room, cancer, right? And the reason why is simply because this is one of the biggest killers in the world. It's responsible for about a quarter of deaths worldwide. And 40% of the people born after 1960 in developed countries will be diagnosed with cancer sometime during their lifetime. At its root, cancer is simply uncontrolled or even uncontrollable cell division. We didn't actually know what really caused cancer at the molecular level until even the 80s. So from the 60s to 70s we thought it was a virus, or several viruses, and it wasn't until relatively recently that we knew that it was a perversion of our own genes. We know now that it is caused by a gradual accumulation of DNA changes, and we call those changes mutations. But it's a little bit narrow, a bit of a narrow definition. Those DNA changes can not only include mutations, they can also include improper DNA methylation, improper histone acetylation, plain old single nucleotide mutations, even entire chromosomal rearrangements. Anything that changes your gene expression can trigger the development of cancer. When we look over here at this graph and we see what are the most common cancer types, one thing that almost all of these have in common, especially if we look at lung, colon, and rectal, and maybe not so much prostate cancer, but especially melanoma, a lot of these are triggered by cells that have exposure to some sort of outside agent. So melanoma is actually caused by a specific cell in your skin called melanocytes. Those melanocytes are what are responsible for producing pigment. And when those melanocytes, let me give you the spelling here, melanocyte, although it's spelled kind of how you think it would be spelled, melanocyte. When those melanocytes acquire mutations, that's when they start growing too fast and become melanoma. So these skin cells are exposed to the outside. They're exposed to UV radiation. They're exposed to all the nasty stuff that you encounter in your everyday life. Same thing with colon and rectal cells. Those aren't necessarily exposed to the outside, but they are exposed to everything that you consume. Same thing with your lungs. They're exposed to everything that you inhale. So cells that are exposed to the outside tend to acquire mutations faster, and so they tend to become cancerous at a higher rate. Those cells that also tend to become cancerous are those cells that divide very often. So the more rounds of cell division you have, the more opportunities you have to make mistakes during cell division, and so the higher the incidence of cancer. So melanoma kind of has two strikes against it. Not only is it heavily exposed to the outside, it is also a product of skin cells which divide quite often. Same thing actually with colorectal cells. So let's look at some of the risk factors for cancer. Risk factors are anything that increase your chance of getting cancer. One of the biggest risk factors is simply age. So the more rounds of division a cell has undergone, the older a cell, the more likely it is it's acquired mutations during DNA replication. Keep in mind that the enzyme that does DNA replication, the main enzyme, remember, his name was DNA polymerase III. And he's pretty accurate. He has that exonucleolytic activity, editing activity, but he still leaves mistakes behind. So cells that are older will have a higher chance of having mutations, and cells that replicate a lot, like all of our mucous membranes. Our mucous membranes are, our nose, our mouth, our digestive tract, our respiratory tract, reproductive tract, all those. They're all of our internal linings. These tend to have a higher rate of cancer as well. So age is the biggest risk factor, but certain lifestyle factors also contribute. Poor diet, lack of exercise. We don't know exactly how lack of exercise contributes to cancer pathogenesis, but poor diet for sure. Being exposed to certain carcinogens. Carcinogens are chemicals that can trigger mutations. Certainly increase your risk of cancer. One thing that is somewhat interesting, especially those of you guys who are in organic chemistry may find this particularly neat. The combination of acid, such as the acid from soda, and the combination of nitrites, the preservative in meat, produces free radicals, and these free radicals will zoom around your cells, and they'll strip off electrons off DNA, off anything that has lots of electrons. So the combination of soda and any preservative-heavy meat will actually trigger mutations. So there's, for quite a while, throat cancer was known as trucker's throat, well, because truckers would stop and get a piece of beef jerky and wash it down with some soda. Now, there are other much more potent carcinogens besides beef jerky and soda. By the way, it's the combination of the two, not not one or the other. That really does it. But anyways, there are other more potent carcinogens besides beef jerky. And one of the most potent carcinogens that we know of is actually cigarette smoke. So cigarette smoke contains a particularly nasty carcinogen called benzopyrene. Benzopyrene causes a pretty severe miscoding, and that leads to an introduction of a ton of mutations. So benzopyrene is no good. And these lifestyle factors account probably for about 30% of cancer cases. Viral infections probably account for about 20% of cancer cases, and we had mentioned those unscheduled S phases. So a lot of viruses will force cells to divide, they'll trigger cells to divide to take advantage of the cell replication machinery. Remember, viruses can't divide by themselves. They have to manipulate, they have to hijack cells' own DNA polymerases and RNA polymerases and ribosomes, so they force a cell to undergo S phase and produce these enzymes. And we call that an unscheduled S phase. Another risk factor is simply pre-existing mutations that you may have inherited from your parents. This is why when you go to the doctor, they'll always do a family history and ask, any relatives who had cancer, because there's a chance they may have passed it on to you. However, most cancers do not arise from pre-existing mutations. Most cancers arise from mutations that are acquired throughout your life. So when researchers really started looking at cancer in the 70s, 80s, and 90s, especially in the 90s as molecular biology tools got better, they started finding out that all cancers share some common characteristics. And when I say all, I should really preface this, I should say most. So these common characteristics, or hallmarks, as they call them, hallmark, it's not just a card store, okay? These common characteristics of cancer are found in almost all cancer types, and it's what allows cancer to be dangerous, these characteristics. So the first characteristic we're actually going to look at is called sustained proliferative signaling. It simply means that a cell will continually either receive signals or make its own signals saying divide, divide, divide. So sustained means to keep going. Proliferative means growth. Signaling, we know what that is. Another behavior that cancer cells will adopt is the ability to evade growth suppressors. So one of the things your cells do to each other is they actually will inhibit each other's growth. This is normal. This is actually good, right? You don't want to overgrow and steal your neighbor's food and oxygen. So cells will tell each other to stop growing after a certain period of time. And cancer cells have actually acquired the ability to ignore those signals telling them to stop growing. Given enough time and given enough opportunity, they will begin to invade surrounding tissues. They will begin to metastasize. Metastasis is simply where cells have broken off from the initial site of cancer formation and moved to another part of the body and reestablished themselves in another part of the body. This one's kind of interesting. Cancer cells will enable what's called replicative immortality. If you remember, when we had talked about DNA replication, we said that every time you do a round of replication, you lose a chunk off the ends of your chromosomes, right? And that if you replicate enough, eventually you're going to eat your chromosomes away into nothing. Although most of our cells don't make it that far. What cancer cells have acquired is the ability to re-lengthen their chromosomes so they can keep dividing and dividing and dividing infinitely. We have cancer cells from the 70s that are still alive and I don't know if you can call them healthy, but they're still alive, they're still replicating, and they will be for as long as we keep feeding them. Cancer cells will also do something called angiogenesis. Angio means blood vessels. Genesis, of course, means creation. They will create their own network of blood vessels. And this is because cancer cells have a high metabolic rate. They need a lot of oxygen, they need a lot of glucose, so they can produce a lot of ATP. So they will create their own blood vessels to deliver those nutrients to themselves. And then, finally, cancer cells will resist cell death. One of the things that happens with cells that go wrong, with cells that acquire too many mutations, is they undergo a process called apoptosis. And I've heard a couple different pronunciations for this. I've heard apoptosis. I like apoptosis, just because it sounds funnier. So cancer cells have acquired the ability to resist apoptosis, and we'll see that in a few slides. Now, there are dozens of different genes that can cause this resistance to cell death. There are dozens of genes that can cause this sustained proliferative signaling. There are dozens of genes that can cause the evasion or growth suppressors, right? What am I trying to say here? What that means ultimately is there are thousands of different possible mutations in hundreds of genes that you can have that can cause these behaviors and that can ultimately cause cancer. When you look at cancer as a disease, it's not really a single disease. It's kind of misclassified as a single disease. It's hundreds, if not thousands, of different diseases. They are all characterized by an overgrowth of cells, though, although they have multiple varying causes. So let's look at that first hallmark, that sustained proliferative signaling. You'll notice right here for you guys I have a link. You can click on this if you have the original PowerPoint up. This is a link, and it leads to a blog post on this. It's a little bit longer than what I have here, actually a lot of it longer, and it really goes in depth as to what's happening here in a very accessible manner. So if you're interested in this sort of thing, I really encourage you to click on that and read through. So let's look at this first hallmark, sustained proliferative signaling. What cancer cells have been able to do is they've been able to speed up the rate of their cell cycle without needing any growth signals. If you remember, most of your cells are in G0, right? Most of your cells are not actively growing, they're being lazy, and they need some sort of kick to get them growing again, to get them to re-enter the cell cycle. And that kick usually comes from growth signals or growth hormones, right? And what oncogenes do, this special set of genes, they allow cells to increase the rate of their cell cycle, and cancer cells really take advantage of that. Cancer cells may mutate these oncogenes, and they may cause these oncogenes to be overexpressed, and these oncogenes may code for growth factors. They may also overexpress a growth factor receptor and respond very strongly to a tiny signal. So oncogenes are any class of genes that can increase the rate of the cell cycle. It can be growth factors, it can be growth factor receptors. Right? The only thing they have in common is that they will increase the rate of the cell cycle. Because of this, cancer cells do not need any external signal to keep growing. So we often say that they have growth signal autonomy. Autonomy means you get to decide for yourself. So they get to grow pretty much whenever they want. To kind of illustrate how this works, I'm going to kind of try and cram this down here in the bottom corner. Here's a cell membrane. Okay. Here's a growth factor receptor embedded in that cell membrane. This is a transmembrane protein. And let's say that this is one of our RTKs. RTK, if you recall, stands for receptor tyrosine kinase. It's one of our major classes of receptors, and RTKs usually trigger cell growth. Well, what a cancer cell may do is it may overexpress the gene that codes for certain RTKs and make lots and lots and lots of these RTKs, which means that even a tiny little signal floating along the outside can cause massive activation and cause that cell to over-respond to that signal. So the cell will divide too much. The other option is simply to have a normal number of RTKs, and also apparently delete your cell membrane too. Let me put my cell membrane back. The other option is to have a relatively normal amount of RTKs, but have too much of this growth factor floating around here. So it achieves the same effect, where the cell responds too strongly and grows too much. One example of an RTK, by the way, is EGFR. Stands for erythropoietin growth factor receptor. Erythropoietin, that's how you spell that. Erythropoietin growth factor receptor. You guys may have heard of that one. Another example where these genes may go wrong is with RAS. RAS is a common oncogene. It's actually a G protein, if you remember that. If you remember way back when, our two major types of receptors were RTKs, or receptor tyrosine kinases, and GPCRs. And G proteins belong to GPCRs. So RAS is a very common oncogene. And there's actually genetic tests that you can take to see if you have particular RAS mutations and to see whether your particular set of mutations will respond to certain chemotherapy drugs. Okay, so let's look at our second hallmark, the evasion of growth suppressors. The major class of genes that's responsible for this behavior are called tumor-suppressor genes. And, I mean, look at their name, right? Tumor-suppressor genes. Hopefully pretty self-explanatory. You may have heard of one tumor-suppressor gene, very, very famous one, called P53. The P stands for protein. Creative naming, right? So these tumor-suppressor genes, as a whole, slow down the rate of the cell cycle. What we saw before with our oncogenes was speeding up the rate of the cell cycle. These slow down the rate of the cell cycle. So think of oncogenes like the gas pedal and tumor suppressor genes like the brake pedal. In order to get a runaway car, in order to get a cell that's dividing too fast, you need to disable both, but in different ways. So tumor suppressor genes like P53 and his buddies, they will detect DNA damage and they will trigger repair enzymes. Now if that damage is too severe, they'll trigger what's called apoptosis instead. And apoptosis is programmed cell suicide. Notice the emphasis here on programmed. This is a method of suicide that has to be orderly, it has to be done in a certain order, and your other signaling pathways have to be intact. That'll come back into play later. This is a very famous tumor suppressor gene, retinoblastoma protein. It was first discovered in retinoblastoma cells actually in children. And it's a tumor suppressor gene that binds to and inactivates something called an E2F transcription factor. Let me give you a little bit of background on that E2F transcription factor first. E2F transcription factors are a group of transcription factors. They are specific transcription factors. And if you recall, you need specific transcription factors to read certain genes at certain times. They help to express cyclin proteins, CDK proteins, DNA polymerase, helicase, all these that are involved in speeding up or suppressing the rate of the cell cycle. So retinoblastoma, what it does is it binds to and inactivates E2F. If E2F isn't active, you can't make these other proteins, therefore you do not enter the cell cycle. Here's actually kind of how this works. So you get some incoming growth factors right there. That was a crappy arrow, I apologize. There we go, that's a better arrow. Not much better, but it works. Those incoming growth factors will trigger the increase in cyclin concentration. And remember, in order to get cyclin working properly, we need to attach the cyclin protein to a CDK protein. And only then do we get MPF, and only then can we enter mitosis. And accidentally switch our slide. There we go. So what E2F does is it binds to retinoblastoma protein. Okay, it binds to retinoblastoma protein. And when E2F binds to MPF, so the E2F, RB, and MPF combination bind together, what that'll trigger is it'll actually trigger retinoblastoma to detach from E2F and the cell to pass its G1 checkpoint. So that's how things normally work. If you mutate RB, if you mutate this guy right here, it prevents it from binding to E2F, and this allows our cyclins and CDKs to be transcribed without any sort of control. So again, if you want to know more, there's that blog post right there. So here's some of our other hallmarks, and I actually think these are more interesting. Immortalization is a big one, and I think on the main slide we had said enabling replicative immortality. It's the same thing as immortalization. I really like this word too. It's just a cool word. With immortalization, cells acquire the ability to divide infinitely. And usually this is because you are overexpressing an enzyme called telomerase. Remember, telomerase is the one that rebuilds the ends of your chromosomes, so you don't eat them away. So it allows for unlimited replication. Now, p53 also has a hand in this guy too, so p53 is not only a tumor suppressor, he also assists in the process of immortalization, or rather, prevents immortalization. Normal, unmutated p53 will actually inhibit telomerase. So if p53 is mutated, he cannot inhibit telomerase, and you can express telomerase. Another hallmark is the ability to evade cell death. Now, keep in mind, we just said this. In order to undergo programmed cell suicide, apoptosis, it requires a lot of time, a lot of effort. Everything has to be in place. Everything has to be perfect, right? So this apoptosis pathway is pretty complicated, and it includes proteins with names like BCL2. And if you mutate the genes for these proteins, well, then the cell can't undergo apoptosis. It can undergo necrosis. Remember, necrosis was plain old decay. But it can't undergo programmed suicide. Because of this, cells that should have died because of a massive amount of mutation keep going. They're kind of like zombie cells. And then cancer cells, given enough time and given enough opportunity, they will also acquire the ability to evade and metastasize. If we recall, this is all the way back from the beginning of the quarter, we looked at a couple different types of ways cells attach to each other. If you remember, cadherins link cells to cells, integrins link cells to their extracellular matrix. Well, cancer cells will mutate the gene that codes for cadherins. They'll mutate the gene that codes for integrins. So those cells no longer attach to their extracellular matrix. And there is other stuff that goes along with this, because most cells that detach from their ECM do die. So this is not the only thing going on, but this is a step. And what's particularly interesting we're finding is that cancer cells can easily switch between turning those linking proteins off and turning them back on when they want to reattach and reestablish themselves elsewhere. In order to do that, they actually turn their genes on and off in a way that resembles a developing embryo. This actually will become a common refrain. Cancer cells look and act a heck of a lot like stem cells. So another common characteristic that cancer cells have is genome instability, and this partly results from resistance to cell death. So this partly results from resistance to, I'm just going to draw a little skull and crossbones here. Okay. What happens here is, because you cannot die as a cell, you are free to keep acquiring mutations. And if you happen to acquire a damaging mutation in a gene that codes for a DNA repair protein, you're kind of screwed. So here's an example, actually. You may have heard of this set of genes, the BRCA1 and 2 genes. Sometimes I hear it pronounced BRCA. The BRCA1 and 2 genes code for DNA repair proteins, and if that DNA repair takes some time, they'll even suppress the cell cycle. So they kind of also act as tumor suppressor genes too. If the DNA damage is too severe, if it's unrepairable, they'll trigger apoptosis. All that contributes to maintaining a stable, mostly unchanging genome. But the problem is, if you mutate your BRCA genes, well, then you lose the ability to repair DNA, you lose the ability to arrest your cell cycle, and you lose a stable genome. BRCA1 and 2 are particularly active in mammary tissue and ovarian tissue, so women with BRCA1 and 2 mutations that disable this DNA repair protein are much more likely to have breast cancer and ovarian cancer. You may have heard of Angelina Jolie and her decision to have a preventative double mastectomy. She had a particularly vicious mutation in, I think it was both BRCA1 and BRCA2, and that led to an 89% chance of her having breast cancer in her lifetime. It's not just 89% increased over normal, it was just an 89 flat % chance of having breast cancer. So she chose to preemptively remove both breasts before that could happen. We do have tests for BRCA1 and BRCA2, but if there's no one in your family who's had breast cancer or ovarian cancer, it's actually not recommended that you undergo the test because your chance of having BRCA1 and BRCA2 mutations are so low. Okay, so another major hallmark is angiogenesis. Again, angio means blood vessels, genesis means creation, and this is where cancer cells will secrete a growth factor called VEGF. VEGF stands for vascular endothelial growth factor. And just based on the name vascular, right, blood vessels, endothelial, these are actually the cells that line the insides of your blood vessels. So when you oversecrete VEGF, you grow blood vessels. So here's an example of tissue that just is growing normally, and here's an example of tissue that has growth, that has this VEGF added. You can see a pretty dramatic increase in terms of blood vessel growth. And tumors do this. They mutate their angiogenesis-related genes, their VEGF gene, really, and switch it from being off to on. So they'll switch it from being inducible to constitutive. And that's just to feed themselves. So, a quick thing about that genome instability, right here. What happens over time is because you cannot repair your DNA, and because you cannot undergo apoptosis, you've lost the ability, you'll accumulate so many mutations, and such severe mutations, that your genome kind of ends up looking like this. So this is a pretty typical cancer karyotype. Remember, a karyotype is the array of all the chromosomes in a cell. And when we look at this, keep in mind that we should really only have 2 copies of every single chromosome, right? Humans have 2 copies of each of their 23 chromosomes for a total of 46. So let's look at chromosome 1 right here. We should really just have these 2. These extra 3 shouldn't be there, so there was a pretty big duplication that happened. Similarly with chromosome 2, I think maybe it was these 2 chromosomes that are supposed to be there. This guy's not. With chromosome 3, we can see that one part of that chromosome actually got broken off, and I think this is actually the broken off part right there. This is pretty terrifying, actually. This is scary looking. And what happens is the more mutations you acquire, the faster you acquire them. Because if you've disabled your DNA repair genes, or your DNA repair proteins, then you're just going to keep snowballing in terms of mutations. So there's as soon as you disable those DNA repair proteins, there's a rapid snowballing mutations due to this genomic instability. And if you can't die as a result of this genomic instability, you're just going to keep those mutations. So that's what leads to these really, really weird looking karyotypes. Okay, so altogether, all these mutations in all these different genes that govern these different hallmarks of cancer, step by step will lead to the development of cancer. Now, cancer development doesn't just happen overnight, at least not most of the time. And we have a model for the development of cancer. It's called the multi-hit model. And in this model, each hit is some sort of mutation. It's a change. All right. And the multi-hit model of cancer says that it takes multiple mutations in several different gene types to develop cancer. And let's take a look at this as an example. Here's a normal cell going about his day. He undergoes some sort of mutation induced by a carcinogen, right? Maybe you drink some soda and eat some beef jerky that day. And that cell he's not truly considered a cancerous cell. He's just growing a little bit fast, all right? And somewhere down the line, one of his progeny will give rise to another cell type, and that second cell type will have acquired a second mutation. And that second cell type, here in the kind of the pink color, will grow yet a little bit faster. Maybe that pink guy will give rise to some purple guys that have a third mutation, and those purple guys can't survive. Their apoptosis pathways are still intact, their lineage is wiped out. But further on down the line, we did give rise to a mutation that does survive, and then that teal guy gives rise to another set of mutations that does survive, and so you end up with a solid mass of cells, some of which carried just that single original mutation, some of which carried, the 1, 2, 3, 4 mutations, some of which carried only 2 mutations. So in a single tumor, a tumor is a mass of cancerous cells, in a single tumor you may have cells with dozens of different genotypes, dozens of different sets of genes with dozens of possible different mutations. This is part of, at least, what makes treatment really, really difficult. You're not battling just one disease in one person, you're battling dozens of diseases in one single tumor type, let alone, if there's multiple tumors. So, now keep in mind that this multi-hit model of cancer takes years to develop. Years and years and years. Let's look at an example now. Let's start out with normal cells. So most of us start out with almost completely normal cells. And these normal cells are living in the wall of your colon. And these cells are exposed to a lot of carcinogens, all the crap that you eat, right? And they divide a lot, so they make a lot of mistakes all the time. So one day, you acquire a mutation that leads to a loss of a tumor-suppressor gene. In this case, it's called the tumor-suppressor gene APC. There's lots of them. Because you've lost a particular tumor-suppressor gene, you don't have the ability to put as hard of a brake on your cell cycle, so you speed up a little bit. Remember, the tumor-suppressor genes are kind of like the brakes, and the oncogenes are kind of like the gas. And then, in that little mass of cells that develops, you may acquire another mutation, and that second mutation, by the way, can come seconds later, days later, years later, even decades later. But that second mutation will come eventually. And that second mutation may activate the RAS oncogene. So not only have you lost your brake, now you've stomped on the gas too. So that little mass of cells is going to get a little bit bigger, it's going to grow a little bit faster. And then, again, decades later, you could get loss of another tumor suppressor gene. In this example, it's DCC. You get a larger growth. Given enough time, and given enough mutations, that cell will acquire the ability to grow its own blood vessels. It'll acquire the ability to invade and destroy surrounding tissues. It'll acquire the ability to resist cell death, and that's when it's considered a malignant tumor. At this point, these are considered benign. The line of demarcation between benign and malignant tumors is when cells from a tumor have acquired the ability to destroy their neighbors. So at this point, we can see those cells are starting to invade into the wall of the organ. That is a malignant tumor. So something to keep in mind, and this is, by the way, this is just an alternate view of what we see here. Something to keep in mind with mutations is that most mutations that we acquire don't do anything. They're neutral, right? They don't help, they don't hurt. And a large part of this is simply because our genome is, well, we think, it's about 95% junk, maybe. As soon as I upload this, of course, people are going to find a use for it, but there's a 95% chance that that a mutation that you acquire is just going to land in an area that you don't use at all, right? Now, of those mutations that do land in a gene or in a regulatory region of a gene, maybe they make a change, but not a big one, right? Now, what about those mutations that land in the regulatory region of a gene that do make a big change? Well, if they cause you to overexpress a tumor-suppressor gene, remember tumor-suppressor genes slow down the red of the cell cycle. If you make too much of it, you stomp on the brake a little too hard, not a big deal, right? So what? That cell divides a little bit more slowly. You have to get the right set of mutations in the right order at the right time in order to develop into a cancerous cell. And part of the reason why we see this is the same forces that drive cancer is the same, I say forces, the same force that drives cancer is the same force that actually drives evolution, and that is mutation. Cancer can be viewed as a set of cells that has figured out how to acquire more nutrients, that has figured out how to divide faster, that's figured out how to survive better in its host organism than its neighbors. So cancer cells are under some sort of weird natural selection to be able to survive better than their surroundings. So mutations and natural selection are the same thing that evolution works with, and it's the same thing that cancer works with too. So it would be very difficult to eradicate the cause of cancer without eradicating our own evolution, or our own ability to evolve. So with that happy note, I leave you. I hope you found this interesting and worthwhile listening to, and thanks as always for tuning in.